Hello, thank you for joining us today. I'm Tracy Smith, the Program Director of Education and Outreach for the Center for Integrated Cellular Analysis based in New York City. In this seminar series, we highlight new papers and methods from our center that we believe to be of interest to the broader community. Today, we will discuss Phospho-seq, a multimodal sequencing-based approach that reveals intracellular protein profiles, as well as cell signaling dynamics and more in single cells. We'll have time at the end of the talk for questions, and we encourage you to either raise your hand and I'll ask you to unmute yourself, or you can type your question into the chat and I'll read it aloud to the speaker. Our guest today is John Blair. John is a postdoc in the CICA lab at the New York Genome Center and received his PhD from the University of California, Berkeley. His research focuses on understanding signaling and gene regulatory aspects of neurodevelopment. With that, welcome, John. I'll hand it over to you.
Okay, thank you so much, Tracy. So I'm here to talk about Phospho-seq, our new method for integrated multimodal profiling of intracellular protein dynamics in single cells. So what is the motivation for this? So why do we actually want to incorporate protein into multimodal single cell assays? So one is that protein tends to be more abundant, so it has fewer dropouts when compared to single cell RNA sequencing. Two, RNA does not always have a linear relationship with protein levels. And then really the main motivation for developing this assay is that we can measure post-translational modifications, such as protein phosphorylation, which are currently impossible to quantify with RNA. And so when imagining this assay and building this assay, we wanted it to not only obviously be in single cells and incorporate RNA and attack, but also we wanted to have several different types of proteins, including surface proteins like cellular hashing marks, nuclear proteins like transcription factors, and cytoplasmic proteins, including those with post-translational modifications. And so when developing this assay, we of course built on the work of others. One key work that inspired this was ASAP-seq from the New York Genome Center, where we use their whole cell permeabilization method. And then also NEAT-seq from Will Greenleaf's lab, where we use their single-stranded DNA binding protein as a method of background reduction. There are still some limitations with these assays in that DNA conjugated intracellular antibodies are not really commercially available. And if you want to order them custom, they're usually prohibitively expensive and they take a long time to be made and then sent to you. And then also fixed whole cell protein quantification suffers from high background, and that also RNA from fixed samples tends to be of lower quality. So with Phospho-seq, we use custom conjugated antibody panels to quantify surface, cytoplasmic, and nuclear proteins alongside ATAC-seq in whole permeabilized cells, and we bring in RNA through bridge integration. So when we're building our custom antibody panels, we use a long-established method of click chemistry to conjugate uniquely indexed single-stranded DNA molecules to whatever antibody of choice. We then take our conjugated antibodies and pool them together, and then that is our conjugated antibody pool. For this method, we did several optimizations so that we can create panels of 100 plus antibodies from most commercial antibodies, and we can do this cheaply and quickly. So this means that whatever antibodies you have lying around in your lab freezer that you might be using for immunofluorescence or flow cytometry, you can most likely use them for a Phospho-seq assay as well, and you can just conjugate them on your own benchtop. When we were testing whether these conjugated antibodies would actually perform well when doing intracellular staining, we used K562 and IPS cells, which are quite different, and stained for the IPS marker SOX2. What we found was that with our unconjugated antibodies, we saw a nice separation of signal, but with our conjugated antibodies, we just saw extremely high signal in both the K562 and IPS cells. Luckily, around this time that we were testing these things, the NEAT-seq paper came out with their method of single-stranded DNA binding protein to reduce the background. And so when we apply this to intracellular staining, we can recapture this signal difference between the K562 and IPS cells when we bind our single-stranded DNA binding protein to our conjugated antibodies. Okay, so with that, we can go through the whole Phospho-seq experimental procedure. So first, you take your cells, you fix and you permeabilize them. Then you block and you hash them with cell surface hashing antibodies. Then you combine all of your hash populations and stain with the single-stranded DNA binding protein bound intracellular antibodies that they're all stained together. Then you input them into TN5 tagmentation and ultimately the 10x single-cell attack sequencing kit. For a pilot experiment, we again returned to the K562 and IPS cells where we wanted to measure GATA1, which is canonically expressed in K562 cells, and OCT4 and SOX2, which are expressed in IPS cells. When we ran this experiment, looking at the attack modality, we saw a really strong separation between the two different cell types, which is expected, but it's also a nice confirmation that our attack sequencing is working well. And then we also saw a strong difference, strong corresponding difference between the cell hashing marks, also showing that the surface protein labeling is working well. If we look a little bit closer at the attack, we can see that at the genomic loci for GATA1 and OCT4, we see open chromatin where we would expect to see open chromatin. And then if we look at the ChromVar scores, which is a measure of transcription factor motif accessibility, we can see that we see strong ChromVar differences between the two cell types as expected. So now, when we look at our actual protein quantification, we can see a very strong GATA1 signal in the K562 compared to the IPS and a strong OCT4 signal in the IPS compared to the K562. So showing that we can do intracellular protein quantification with high specificity from fixed permeabilized whole cells. So next, we wanted to make sure that we could also quantify phospho and cytoplasmic proteins. So we took our K562 population and treated one half of them with EGF to stimulate the PI3K AKT mTOR pathway, and then another half with PX866 to inhibit the same pathway. For this experiment, we used phosphoribosomal protein S6 as a readout. What we found was that in the stimulated cells, we had a much higher signal of phospho-RPS6 compared to the inhibited cells. And this was not merely just a reflection of total RPS6 levels. So with this, we can show that cytoplasmic proteins and post-translationally modified proteins are indeed quantifiable with Phospho-seq. So next, we wanted to apply Phospho-seq to a more heterogeneous cell population and also focus on something that was of more biological interest to myself. And in that case, it's neurodevelopment. And so for this, we collaborated with Barbara Treutlen's lab at ETH Zurich, who kindly donated 3-month-old brain organoids to us so that we could run Phospho-seq on them. In this experiment, we used a broadly expanded antibody panel with many more phosphorylated proteins, canonical transcription factors, and also cytoplasmic proteins. When we look at the attack-seq for this experiment, we can see lots of different clusters of cells, indicating that, again, the attack sequencing worked and that there is a high amount of heterogeneity within this data set. If we use the chromatin accessibility alone, we can create these rudimentary cell type labels for things that encompass different lineages and multi-regions within early neurodevelopment. If we look at within these cell types, we look at nuclear proteins such as SOX2 and OTX2. We see that we see a strong difference between different cell types that also closely matches the chromatin accessibility at the respective genomic loci. This is also the case for cytoplasmic proteins. But we want to be able to bring in RNA. And so for this, we turn to a recently developed technique from the CICA lab, bridge integration. And so our data set right now is an attack-seq plus protein data set. That's our Phospho-seq data set. And what we can do is we can include 2 extra data sets. One is a multimodal bridge that has RNA and attack, and another is a high-quality RNA data set that was run on unfixed whole cells. And so with bridge integration, you find anchors between the different modalities such that you can bring the RNA from the high quality reference all the way over to your attack plus protein data set, giving you a final trimodal data set of attack, protein, and RNA. We can use the RNA first to just increase the cell type labeling, to increase the specificity of the cell type labeling such that we get more specific cell types. And so then what can we actually do with a final trimodal data set? So we can learn how differentially active cell signaling pathways affect chromatin accessibility and gene expression. We can reveal protein-associated gene regulatory elements that affect proximal RNA expression. And we can determine relationships between developmental gene expression, protein expression, and functional activity of protein. So focusing on signaling first, we wanted to find differentially active signaling pathways in different cell types. And so what we observed in this data set was that there was an increase in phosphorylation of members of the MAP kinase ERK pathway that we observed in diencephalic cells, or cells from the diencephalic lineage versus cells from the telencephalic lineage. So this was interesting unto itself. But also the MAP kinase ERK pathway is specifically interesting because one of the terminal phosphorylation events of this pathway is phosphorylating ribosomal protein S6 through this kinase RPS6KA1. However, RPS6 also gets phosphorylated through activity of the mTOR pathway through RPS6KB1. So this led to a question of, were the differences in phospho-RPS6 signal that we are seeing in our cells driven by MAP kinase ERK, as we might expect, given the increase in MAP kinase ERK signaling that we see, or is it being driven by mTOR? And so for that, we returned to chromatin accessibility, where we just looked at the chromatin accessibility of both RPS6KB1, which is, again, controlled by mTOR, and RPS6KA1, which is controlled by ERK. And we looked at the open chromatin as it related to phospho-RPS6 levels. And what we found was that for RPS6KB1, the open chromatin didn't really change, while for RPS6KA1, there was more chromatin accessibility as the phosphoribosomal protein S6 signal went up. So this strongly indicates that in this lineage, the differences in phospho-RPS6 signal are being caused by the MAP kinase ERK pathway. So this is just one specific example of how quantifying signaling pathway activity alongside chromatin accessibility can connect gene regulation to cell signaling. So now, if we look at transcription factor quantification and motif accessibility, we can look at the levels of transcription factor protein and see that they look pretty similar to the ChromeVar scores for those respective proteins. And so not only do they look very similar, if we look at it in a different way with rank motif correlation, we can see that these are actually at the top hits. So the cells with high OTX2 protein are the most correlated transcription factor motif is OTX2 itself, and the same goes for SOX2. So given that observation, we figured that we could also use this type of analysis to identify different transcription factors that are associated with non-transcription factor proteins. For instance, if we look at the levels of phospho-RPS6, we identified that the hippo pathway-associated TEAD factors are strongly associated with phospho-RPS6 signaling, and I'll touch more on this in a few slides. With the transcription factors, we want to be able to identify functional gene regulatory elements as well. So using OTX2 as an example, what we can do is we can look for open chromatin that is highly correlated with OTX2 expression. So if we do that using a fairly stringent cutoff of 0.5 for open chromatin and protein, we can find about 5,000 peaks that are highly correlated with OTX2. We can then filter those peaks for those that actually possess an OTX2 transcription factor binding motif, and this lowers it to about 2,500 highly correlated peaks with the OTX2 motif. If we take these highly correlated peaks and do a footprinting analysis, what we observe is that there's a high TN5 insertion bias surrounding the OTX2 motif in cells that have really high OTX2 expression, and not in cells that don't really express OTX2. If we look at the peaks that are not correlated with OTX2, we can find that this effect mostly disappears. So with this, we can be fairly confident that what we're actually observing are true OTX2 binding sites that might actually be bound by OTX2 at the time of running the assay. So now we want to bring this and tie this into RNA. And so with RNA, we look for RNAs that are correlated with our protein expression and also proximal to our highly correlated peaks with OTX2 motifs. And when we do that, we then get a series of peak gene links that we can separate into two bins. One is an activating bin, activating peak gene link, where as chromatin becomes more accessible, OTX2 binds or other transcription factors bind, and the proximal RNA increases in expression. Or you can have a repressive peak gene link where the opposite happens. As the chromatin becomes more accessible, the expression of the RNA goes down. If you take lists of these for each transcription factor that we've profiled in our assay, you can start to build gene regulatory networks where we can identify genes that are being co-regulated by different transcription factors, including cooperatively, and also antagonistically. So now, returning to the signaling to gene regulation relationships, as I mentioned before, we found that phospho-RPS6 is highly correlated with the TEAD factor activities. So what we wanted to do was then look for peaks that were highly correlated with phospho-RPS6 signal and filter them for those that possessed a T1 motif. With that, you can find many examples, including this one as Gas7. You can see that the RNA for Gas7 goes up as this peak becomes more accessible. So then when we take this list of peaks and we submit it to gene ontology, we can find terms like cell division and regulation of cell morphology. And so cell division and cell morphology have been long associated with phospho-RPS6 activity. So this is just an example of how quantifying signaling pathway activity alongside chromatin accessibility can connect the signaling to transcription factor binding and downstream causal effects. So another thing that we want to look at is the RNA-protein-chromatin relationships. And so because we have a developmental data set, we can actually look at this in a fairly interesting way. So for this, we subset our data set into just the telencephalic lineage, going from 4 brain precursors into neurons. And we aligned these cells along a pseudotime axis and then separated them into 3 different bins of early, middle, and late. If we look at, for example, SOX2 and its expression along these, we can see that from the early to middle transition, the SOX2 RNA starts to get shut down, and the quantity of SOX2 RNA goes down as the development continues. But because there's still residual SOX2 protein that was already made by previous SOX2 RNA, that level stays fairly constant during this middle transition. And because the SOX2 protein is the actual active biomolecule, we see that the SOX2 motif accessibility stays fairly constant and actually goes up a little bit during this transition. As the cells transition from a middle to a late stage in development, we see that the SOX2 protein starts to go down, and we see a corresponding decrease in the SOX2 motif accessibility. So if we look at just the rank motif correlations for the ChromeVar score for SOX2 and with protein, we can see that it's fairly high in the early, middle, and late stages. But if you look at the RNA, the correlations are actually not that high for the early and middle stages and only stabilizes during the late stages. So this is an example of an interesting dynamic that you can look at when you have a developmental data set with Phospho-seq, but also it's a really strong example of why you actually want to profile protein as opposed to RNA when looking at developmental transitions, because protein is the molecule that's actually performing the activity, and RNA is sort of we use as a proxy for protein. Okay. So then finally, just want to show that we can also do Phospho-seq in other tissues. So we can perform it in vitro hematopoietic stem cells or 34-week retinal organoids. And these experiments were performed with very different antibody panels that we were able to custom conjugate quite quickly. So whatever tissue you're interested in, you could run Phospho-seq. And so then finally, just want to announce and indicate that we're creating, in the process of assembling, a Phospho-seq starter kit. And so I have the box right here. And so what this starter kit will include material to conjugate 4 separate antibodies that you might want to ready for input into a Phospho-seq experiment. So it will contain readily labeled, uniquely indexed oligos. And then you just need to go into your freezer, find whatever antibody you might be interested in, pull that out, and then you can label it with those oligos and you can run your own Phospho-seq experiment. So these kits aren't quite ready yet, but please follow at SEGS ICA on Twitter for more information in the coming weeks about how to get these kits. And so with that, I'd just like to summarize and thank you for your attention. So here I presented Phospho-seq. which is a method for simultaneously profiling chromatin accessibility and intracellular proteins in single cells, where we bring in RNA through bridging to create a trimodal data set. We can create large intracellular panels very cheaply and easily with antibody conjugation. And then in this paper, we applied Phospho-seq to 3-month-old brain organoids to discover gene regulatory and cell signaling patterns during early development. So I'd just like to acknowledge the members of the CICA lab, particularly Carol Delgarno and Austin Hartman, for helping me with all of this work, and also members of the Troitland lab and the Innovation Lab for their strong contributions to this work. And I'd be happy to take any questions now. Thank you.
Wonderful. Thank you so much, John, for sharing your work with us. So as John said, we'll move on to our Q&A. If you'd like to ask a question, you can raise your hand or type your question into the chat box. Here's our first question. What kind of resolution does it have?
Do you mean by like cell type or cell number resolution or?
Yeah.
Okay, well, for cell numbers, basically because in this iteration of the assay, we're using the 10x kits. With hatching, you can push these up to 20,000 cells or something like that, but you can run as many lanes as you like. And then in terms of resolution for different antibodies, you can use, we haven't really hit a limit yet because the indexed single-stranded DNA oligos, the index allows you to have so many different combinations that for what we've run a panel of over 100 now, and we had no problems with that quantification.
Oh, that's great. Thank you. Another question here in the chat. Which CICA antibody platforms have you used? Example, 5'.
Oh, I haven't used 5' CICA antibodies, but we have done a few experiments where we did include other CICA proteins, other CICA antibodies or antibodies that are compatible with CICA. I forget. We used CD55, and then I can't quite remember which other ones that we used, but also the hashing antibodies are CD298 and B2M. So everything we've seen thus far is fully compatible with the CICA antibody panel, but I can't speak to whether the 5' would work well with this assay.
Okay. Okay, a lot of questions in here. For the gene protein-chromatin, sorry, things are moving. Oh, and thank you for sharing that. Sorry. For the gene protein-chromatin analysis, how did you assign the pseudotime access to the data?
Right. So with the pseudotime, we use Monocle for assigning that. And then for looking for the middle or for the early, mid, and late bins, I actually assigned those where there were small clusters of forebrain precursors that were sort of transitioning towards the neurons. So basically, the early is forebrain precursors that are likely self-renewing. So they're sort of staying in their forebrain precursor state. And then there was a section that was closer to the intermediate precursors and then the neurons that through their gene profile were actually in a transition, as you can see with the SOX2 decreasing. And then late was just the intermediate precursors and the neurons.
Okay, thank you. It's another question. How is the relative protein abundance estimated? i.e., is the binding efficiency of the different antibodies taken into account?
Oh, that's a great question. So with this assay, it's very hard to estimate the binding efficiency of the antibodies. Every single antibody is different. And so within this assay, you have antibodies that work really well and antibodies that don't. And so when you're quantifying these proteins, it does have to be relative to other cells within the assay. I wouldn't suggest using this and saying, okay, this cell type was really high, or this cell type was high for this marker in this assay, and then in a parallel assay that I ran, it was lower in a different cell type, therefore we can compare the two. So everything, unfortunately, does have to be relative within the assay itself. But yes, we can't really take into account at this time the antibody dynamics, but as we do use more antibodies and move forward, we'll be able to pare down a list of antibodies that work really well in this assay and those that don't. And so when we have our big panel of antibodies that I used here, not every single one of them worked well. And that's a really important point. You could sort of, in these early experiments, we're using it somewhat as a screen where we can find ones that do work well, put those aside, say these ones are good, and then the other ones we most likely won't use again.
That could be interesting in the future when people use the Phospho-seq kits for people to share what antibodies they used. And
absolutely. And I haven't had time yet or the expertise. I desperately want to build a common resource that people can look at the Phospho-seq data that I've run and others have run for guidance about which antibodies work the best.
Wonderful. If anyone's interested in collaborating with it, I'm sure, as we hear, John is desperate for help on that. We'll take one last question here. Can you distinguish the phosphorylation of different residues on the same protein? That's
a great question. So if you have highly specific antibodies for both of those residues, yes, because the antibodies themselves are uniquely indexed. And really we're taking, when the antibodies are actually binding to the protein, it's not binding, they're not all binding to the same protein. There's so many copies of these proteins that the antibodies are likely binding to different ones. We're sort of just taking an overall a level of the phosphorylation. So yes, you will be able to do that if you have specific antibodies for those two different residues. So when I say phospho-RPS6, in this case, I'm really referring to phospho-RPS6 at serine 235, 236. But if you want to look at 240, 244 as well, which I know there's another highly specific antibody, you could include that in the assay and that would work.
Great. Okay, one more question, because so many great questions here. Have you looked at how the cell surface staining distribution for permeabilized cells compared to intact cells?
Cell surface staining.
Yeah.
So all the cells in here are permeabilized, and we use a control that I didn't talk about to ensure that. And so what I do is I include just a little oligo that has no tag on it. And this is actually something, this is why you tend to have high background with intracellular protein without the single-stranded DNA binding protein, is that if you just put DNA into a permeabilized cell, it will just stick wherever because DNA is negatively charged and it will find some sort of positively charged pocket and just stick in there, and then you'll end up sequencing that. So the single-stranded DNA binding protein masks that signal, so that's why we can reduce the background. But so what I do is I include just a couple of different just pure oligos, and then I use that as a permeabilization marker. So cells that have levels or comparable levels of those oligos in the sequencing, I can say are permeabilized. And then we do have very, very, very few cells where we don't see any reads for those, or very, very low reads. And those we can say are unpermeabilized, and then we just throw them away. But I haven't, to answer the question more specifically, I haven't actually looked at the differences for cell surface protein levels between permeabilized or unpermeabilized cells. I think, running a parallel CICA experiment with something like this would be really informative for that, but I haven't done so. But I would imagine that the signal for the permeabilized cells would be lower, but it should be enough for you to make confident calls about what cell types you have based on those surface antigens. Yeah.
Great. Well, thank you so much, John. Thanks for this great discussion, and thank our whole group here for wonderful questions. So to close out here, the seminar has been recorded and it will be available soon on YouTube and on our website, which is multimodalintegration.org. And you can also follow us on Twitter at segs_ICA, or check our website for the latest announcements regarding upcoming talks and events. And with that, I'd like to thank everyone for joining, and we hope to see you at our next seminar.